Good afternoon, everybody. Welcome to Asphalian's webinar, Biological Impurities, Scientific and Regulatory Aspects. Before starting, just a couple of things to take into consideration about the webinar today. It will last approximately 30 minutes, including Q&As at the end. In case you have any questions, please submit them in the chat. We will try to reply them at the end of the webinar. Otherwise, we will reply them by email. We'll also send you the presentation per email, as well as the link to the recording next week. And now it's my pleasure to introduce you our speaker today, Mario López de la Cruz, Scientific and Regulatory Affairs Senior Consultant. Mario has a degree in pharmacy and 2 master degrees on the fields of pharmaceutical industry and health technology assessment and market access. He has extensive experience in CMC aspects for chemical and biological products focused on the US and EU regulatory framework. In today's webinar, he will guide you through the scientific complexities and evolving regulatory landscapes surrounding biological products. Before that, a very short introduction from our colleague Carlos, who will briefly introduce you our CMC services. Carlos, please.
Hi, Lydia. Thank you so much for your introduction. First of all, I would like to say that we are happy to announce that Asphalion is in its anniversary this year. We are 25 years in the area right now, so we are really happy to share this with you. And please keep our link pages, so more to come about our anniversary. And now I would like to showcase briefly our CMC services. As you can see, we can provide end-to-end support during the whole product life cycle, starting in drug discovery, aiding you with a regulatory roadmap, feasibility assessment, and making ready that you are up to the clinical trials. Also, we can provide support within all the clinical phases with all CMC aspect, writing of the IMPD, interaction with the different agencies, and also dossier preparation and obviously market authorization and post-authorization. We can provide gap analysis services, due diligence, strategic consulting, CMC writing, and more on that. And just briefly to say that everything we do for CMC, we also do for medical devices, and we also have clinical and non-clinical experts and pharmacovigilance experts that can also aid you through your journey. Thank you so much.
Thank you, Carlos. And Mario will take it from here. Mario, please, whenever you are ready.
Okay, thank you, Lydia and Carlos, for the introduction. First of all, welcome everybody to this webinar that will be related to the scientific and regulatory aspects of impurities in biological products. This is a brief agenda of today's webinar. We are going to provide an introduction to what a biological drug is. We will follow with a review of the main applicable legislation specific for the EU and US territories. And then we will share some general concepts that can be obtained from this legislation. And then we will focus on what matters in today's webinar, which are the impurities in biological products. And as Lydia said, we will finish with a section of question and answers if we have time. Okay, so maybe the best way or the best approach to start this webinar is defining what is a biological product. In this sense, this may vary from one region to other. And in today's webinar, we will focus on Europe and the United States regulatory framework. So when we consult the current legislation in Europe, we can find that a biological product is a biological substance obtained from a biological source, and it's a product that for its proper characterization is required to apply an extensive set of and combination of physicochemical biological tests. Okay. Then when we consult the current legislation in the US, we will find that a biological product is defined as a wide list of products. We can find virus, toxins, antitoxins, vaccines, etc., and so on, a big list of type of products that are applicable to the prevention, treatment, or cure of a disease or condition of human beings. We could say that definitions are very similar, but before moving to next slide, we would like to highlight a definition that is present in the US legislation, and this is related to proteins. In this sense, a chemically synthesized protein is a protein that is manufactured by a chemical manufacturing process, synthetically, but which have amino acid sequence not larger than 40 amino acids. In this sense, when we are developing or we are working with a peptide that has a amino acid sequence larger than 40 amino acids and or is manufactured using a biological process, this has to be registered as a biological product. However, the way to know which is the proper regulatory procedure to register a protein, it's not so important the length of the amino acid sequence, because it is important, but it is more relevant which is the manufacturing process used to manufacture this molecule, which if it is a synthetic way or if it's a biological process. Okay, so as we have just seen in the definition in the US framework, we can find under the scope of biological products a wide range of products. We can find any biological substance from human or animal origin. We can find also small biologics, such as peptides. We can also find large biologics like antibodies. We will also find vaccines and immunotherapeutic products, and also the so-called advanced therapy medicinal products, the ATMPs, fall under the scope of medicinal products, biologic products. And within this group, we will find gene therapy, somatic cell therapy, and tissue engineered medicines. Okay, now that we have what is a biological product, we are going to briefly review which characteristics are typical in this type of products. The first of all, these are considered complex molecules. When we compare biologics to the traditional chemical entities, we can see that the molecular mass increase, and therefore this implies a complexity in the structure of these molecules. This complexity also implies the development and the use of sophisticated analytical methods to properly characterize these substances. The thing is that some assays used to test and control these products sometimes present high variability, and therefore it's very critical to define a proper set of specification and analytical methods to be able to discriminate the desired product from the potential variants or forms that can be present in the product, because these forms or variants may have severe side effects. And also due to the source that we are using to manufacture this type of products, biologics has present an inherent a potential of immunogenicity. Sorry. Okay, more characteristics. These type of products are typically less stable than the traditional chemical entities, and this is due to its nature. When we were defining what is a biological product, we share some ideas about proteins, and as probably the audience know, proteins are impacted by some conditions like the temperature, oxidation, mechanical stress, and so on. Therefore, these molecules are considered less stable than traditional chemical entities. Derived from this characteristic, when we are manufacturing sterile biological products, the way to sterilize our product will be basically based on aseptic conditions, because other sterilization processes like thermal heat or radiation will not be able to be applied in the manufacture of biological products. Another thing important is that well, since the source to manufacture these biological products sometimes are cells, microorganisms, and so on, which at the end sources with an inherent heterogeneity, the product that we will obtain using these sources also will have an inherent heterogeneity. And another thing is that the manufacturing process of this type of products, it's very critical. It's very important to develop and have a deep knowledge of the manufacturing process that we are using in order to obtain a batch-to-batch consistency. Because we are not talking about, for example, reference product and biosimilars. No. The thing is that inside the reference product and inside the biosimilar, we will find a small variability from batch to batch. And this will be defined according to the manufacturing process that we are applying. In this sense, when we talk about traditional chemical entities, we could say that the specification defines the product, but when we talk about biological products, the manufacturing process defines the product. Okay, now that we have an idea of what biological products are, and we are going to briefly review the applicable legislation and following the same approach than with the definition. We will focus on Europe and the United States. Due to the characteristics that we have just reviewed, it has been required to develop a specific guidance for biological products. It is true that the traditional guidelines that are used to develop and manufacture chemical entities, the principles described in this guidance are also applicable to biologics, but due to the complexity of biologics, it has been required to develop specific guidance. Also, we will find that this type of products has specific regulatory procedures different to the chemical entities. And related to what I have just said regarding the manufacturing process, and from a life cycle maintenance perspective, at any time that we are applying a change in the manufacturing process and we are preparing a post-approval change in our approved product, it will be required that the applicants perform a comparability exercise between the pre-change and post-change product in order to assure that those products are comparable. And the change doesn't have any impact on safety, efficacy, or quality of the product. In this sense, the majority of the variations, post-approval changes that will be requested in the European framework will be classified as type 2 variations, and prior approval supplements in the US framework. So, well, as said, we will focus on the EU and the US, but as probably the audience know, the principles and the guidances described and issued by the ICH has to be considered. Because this organization, the purpose of this organization is to harmonize the application of technical requirements in medicinal products, and therefore they are like the gold standard to be followed. And in this sense, from the ICH guidance, we will obtain the main principles, and then in the local legislation in the Europe and the US, we will find more specific regulation. Okay. As said before, there are guidances applicable to both to chemical and to biological products, which might be the Q2 guideline related to validation of analytical products, and also the Q11 related to the development of manufacture of drug substances. But we can find specific guidelines for biological products. In this sense, we will find the Q5 series and the Q6P related to the specification. We will talk more about the content of these guidelines. And well, when we consult the EMA database or web page, as said before, we will find more specific regulation. In this sense, we can find a specific guidance on the control of DNA and household proteins, which are impurities. We will talk about this later. We can find a specific guidance for a specific type of products like monoclonal antibodies or cell-based medicinal products and so on. And we will have the same situation if we consult the FDA website or database, more specific guidance. So now we are going to take these guidances and extract the most relevant information. We will start with the ICH package, and the first one will be the Q6P related to the specifications of biological products. This guideline helps us to define the analytical methods and the acceptance criteria that should be defined for our biological product. The first thing that this guideline, the first idea that this guideline share is something that we have mentioned before in the characteristics of biological products. Since we are working with cells or microorganisms, which are organisms with a inherent heterogeneity, the product that we will obtain also have an inherent variability and heterogeneity. Therefore, this is something that should be properly characterized in order to maintain a batch-to-batch consistency and to proper differentiate between the desired product and the potential variants and forms that may arise during the manufacture or the storage of our product. In what regards to impurities, well, this guideline provides a preliminary classification of impurities that can be found in biological products. We will have two types of impurities, which are the product-related impurities and the process-related impurities. Sorry. The product-related impurities are those variants or forms that arise or may arise during the manufacture or the storage of our biological product. And the process-related impurities are those derived from the sources used to manufacture our product or also related to the downstream steps that we have defined in our manufacturing process. We will go deep inside this later. Before moving to the next section, we would like to highlight one thing related to the product-related impurities that this guideline share with us, and is that in the case of variants and forms with comparable properties of the desired product, this will not be considered as product-related impurities, but as product-related substances. This is something that, from a regulatory point of view, should be considered. And another section that is included in this guideline are the contaminants. These are not considered impurities in the sense of how are considered the product and process-related impurities, but the potential contamination with this contaminant must be considered for all the manufacturers. And these are all advantageously introduced materials not intended to be part of the manufacturing process. Okay, so what we can find within the product-related impurities and the process-related impurities. In what regards to process-related impurities, which are those derived from the sources that we use to manufacture our product, we find the potential origin of these impurities are cell substrate derived impurities, cell culture derived impurities, and downstream derived impurities. From cell culture derived impurities, we can find proteins derived from the host organism, from cells, for example, or from the other microorganisms, and we can find nucleic acids. From the cell culture derived impurities, we will find inducers, antibiotics, serum, and other media components. And from the downstream perspective, we can find enzymes, chemical and biochemical processing reagents, inorganic salts, leachables, solvents, ligands, and so on. In what regards to product-related impurities, we can find truncated forms, which are hydrolytic enzymes or chemicals from the cleavage of peptide bonds. We can find other modified forms like diamyrated, isomerite, mismatch, disulfur bonds links, etc. And also the aggregates, which are the dimers and higher multiples of the desired products, would be considered as product-related impurities. Moving to the next ICH guideline, now we will review the Q5 series that we will find information related to different aspects of biological products. The first one is related to viral safety evaluation in biological products. And well, as said before, due to the sources that we usually use in the manufacture of biological products, there is a potential risk of viral contamination. Okay, and the sources of this contamination might be from the cell substrates, the cell lines, and also there is a risk from the exogenous introduction during the production. In what regards to the cell lines, the risk of viral contamination may come from the derivation of the cell line from an infected animal. Also, when we are selecting the cell line and in the process we use virus. Also, when we use contaminated biological reagents or raw materials, and also during to the handling of the cell banks. From the exogenous contamination perspective, well, the potential sources of contamination is the use of contaminated biological raw materials or reagents, contaminated viruseed, contaminated also materials or reagents during the downstream steps. Also, if we use contaminated excipients during the formulation, or just from the environment. If we do not develop a proper aseptic condition for our manufacturing process, there will be a high potential risk of contamination. And this guidance propose three approaches to reduce the risk of contamination of viruses, which are to develop a proper clearance capacity in our manufacturing process, also the proper selection and testing of raw materials and cell lines, and also developing a proper set of controls in both in the manufacturing process, but also in our product. And before moving to next guideline, we would like to highlight that this guideline also say that no conventional agents like prions will not be covered by the principle described in this guideline. So moving to next guidance is from Q5 series. We will find the Q5C related to the stability testing of biological products, and also the Q5D related to the characterization of cell substrates. In what regards to impurities, these guidelines don't provide any different information from what we have, from the ideas that we have shared so far, but they only reinforce the idea of the impurities. For example, in the stability testing guidance, they define an impurity like any component of the drug substance or drug product which is not the chemical entity defined as the drug substance, an excipient, or other additives to the drug product. Another point that make effort the guideline is that due to the complexity of biological products, it's not possible to define the purity of these products based only on one analytical method. Therefore, it is required to follow an orthogonal approach and I use a set of different analytical methods to properly characterize and control our biological products. And the Q5D guideline provides guidance on the characterization of the master cell bank or the working cell banks, if applicable, which basically are our raw materials. The last ICH guideline that we will talk today is the Q11 that we have said that this guideline is applicable for both chemical entities and also biological products. And well, we can find general ideas like the proper definition of the critical quality attributes. This is something that should be considered also in biologics, and also provide guidance on the proper selection of the starting materials. When we talk about biological products, our starting materials will be the master cell bank or the working cell banks, and also the other raw materials that we are using during the manufacturing process. In what regards to impurities in biological products, these guidance do not provide any relevant information. They provide the classification of impurities that we have just seen in the previous slides, that we can find product-related impurities and process-related impurities. So now we are going to briefly mention some ideas in a specific guidance from the EMA, the European Medicine Agency. And the first one is related to these process-related impurities, which are the DNA and the host cell proteins. So these guidance say that the HCPs, the host cell proteins, have to be tested always on routine basis. It's not possible to apply any skip testing or other approach. It is required to be tested on routine basis. And the DNA have the same situation only if we are manufacturing our product derived from continuous mammalian cells. So the approach that we have to follow when facing these two impurities is first to follow a validation approach to properly establish the proper limits of these impurities with the good level of these impurities to be administered to patients, but also it is required to have a routine approach, developing and applying different set of specific controls, both in the manufacturing process, but also in our biological product. This guideline also say that for residual DNA, the routine testing is not required for products derived from bacteria and yeast, and also reinforce again the need to test on routine basis the residual host cell proteins due to the potential immunogenicity of these impurities. We have here another guideline. In this case, is related to monoclonal antibodies, and in this case, they also reinforce the criticality of properly controlled process-related impurities such as host cell proteins. And the reason for this is that this parameter is highly dependent on the process. Okay. And now we would like to finish today's webinar with mentioning some specific situations with some type of products, and these are products which are under development. We can find both in the EMA and also in the FDA specific guidance for products under development, for IMPDs and for IND in the US. And well, just the principles are the same, but this guidance and guidelines may help us to in what regards to the level of detail that applicants should include in both in the IMPDs and the INDs. And finally, we would like to mention that other products like biosimilars have specific guidelines issued from EMA. And well, it is important to say that biosimilars are not generics of biological products. This is other things, and because generic is like exactly what the reference product is, there are some difference, but in what concerns to the molecule, it's the same. But this is something that, from a biological product perspective, cannot be applied because, as we mentioned at the beginning of the webinar, there is an inherent variability from batch to batch of the same product. Therefore, biosimilars are products with minor differences compared to the reference product, but these differences do not pose a clinical, are not clinically meaningful. And also biosimilars within the application to any regulatory agency are required to provide scientific evidence that any aspect of safety, efficacy, of quality is not affected compared to the reference medicinal product. Well, this is something that we have just said, and which differences are allowed in terms of biosimilars? Well, there might be minor variability, for example, in terms of glycosylation. There might be differences in terms of formulation, the pharmaceutical form, and also in terms of administration device. Well, at this point, we just would like to thank you, the audience, to join to this webinar. I hope it has been helpful for you and that this have complied with your expectation.